Good afternoon, everyone. My name is Deirdre Griffin LaHue, and I'm an assistant professor of soil health and sustainable soil management at the WSU Northwestern Washington Research and Extension Center in Mount Vernon. And I'm going to be talking with you today about a project that's been going on long before I started at WSU. It started in 1996, but we're looking at the effects of long-term biosolids applications on soil health in dryland systems of Eastern Washington. And I'm working on this project with Dr. Andy Berry, who is located at the Puyallup Research and Extension Center. So soil health assessments have become increasingly common, and these assessments integrate biological, physical, and chemical indicators or properties of soil that we believe to be important for building productive and resilient agricultural systems. And also looking at how these different properties interact. For example, how microbes break down organic matter inputs that they receive from various management practices, and how this breakdown and carbon cycling can lead to improvements in soil structure, building better structure. But of course, soils are incredibly diverse, and our cropping systems are also incredibly diverse. So it's sometimes a challenge to apply these frameworks to different systems, particularly in the Western US, where there's been less research overall compared to, for example, the Midwest or the Northeastern US. So in each of these systems and in my research, I always like to think about what is the biggest soil challenge in a particular system or environment, or what is that system's biggest barrier to being as resilient and productive as it can be. So for this particular system, we are in Douglas County. As I mentioned, this is a long-term biosolids experiment. We call it GP17. This site gets about 10 inches of precipitation a year. So, you'd think right away that available water or water holding capacity could be a big challenge, particularly in the coarse soils that are in this site. So these are sandy loam soils. And as I mentioned, this experiment started in 1996. So since then, every 4 years, biosolids have been applied to these soils at different rates. And so the treatments, as I mentioned, there's different levels of biosolids. So we have 2, 3, and 4.5 dry tons of biosolids per acre. And then there's both a no fertilizer control and a synthetic fertilizer control with using anhydrous ammonia. So the research questions that we set out to answer were, how have long-term biosolids applications impacted physical, biological, and chemical soil health properties in this dryland system? And also, how do these properties vary with depth? As I mentioned, this was transitioned to a no-till system where the biosolids are surface applied. So, looking at the depth profile in the soil, how do these effects on soil properties differ? So a lot of the data I'm going to be showing you today is actually from the Soil Health Institute's North American project. We were lucky enough to be one of the sites selected for this project up there in the left-hand corner. And these sites were sampled in April of 2019. And this was right before a biosolids application. So the last time biosolids had been applied at the time of sampling was in 2016. So I'll jump right in with some results. Looking at physical properties, here we show available water holding capacity, which, just as a reminder, is the difference between field capacity and the permanent wilting point. So it's the water held in the soil between those two water potentials. And we found, I'll walk you through these graphs because they're going to be similar. In gray, I have the no fertilizer control. In black, we have synthetic fertilizer and anhydrous ammonia. The lighter blue is the 2 dry ton per acre. The medium blue is the 3 dry ton, and the darker blue is the 4.5 dry ton. And so here we're showing the volumetric available water holding capacity. And you can see that particularly at the higher 3 and 4.5 dry ton per acre rates, we see a significant increase in available water holding capacity, which in this rain-fed system with low rainfall is pretty important. We also see decreases in bulk density with all of the biosolids treatments. So showing that there's less compaction, and this is probably also contributing to greater porosity availability for water to be held in the soil. So I do want to mention that there have been scoring curves developed largely by the Cornell Soil Health Framework, the comprehensive assessment of soil health done by Cornell. And so I've plotted our points for available water holding capacity here on their scoring curve for the coarse soils. I have converted these from volumetric to gravimetric available water holding capacity. So if the values aren't exactly the same, that is why. But you can see here that both of the non-biosolids treatments are in the medium zone, the medium score zone. So they kind of get an average score. As we get into the 2 dry ton per acre, we're getting into the green, and these higher treatments with the 3 and 4.5 dry ton are in the very high score. So we're really having an effect here on this coarse soil in terms of available water holding capacity by adding biosolids. So I want to move on now to the biological soil health indicators. And the ones I'm going to be talking about are really kind of chemical indicators of biological activity. So we have soil respiration, which is an indicator of microbial activity, the amount of CO2 being respired by the microbes as they break down organic matter. We're also looking at the active carbon pool, which is thought to be microbially available carbon. It's the amount of food that's available for microbes to break down and turn into more stable organic matter. And then also the soil protein pool, which is kind of a similar biologically available nitrogen pool. So jumping in here to the carbon pools, on the top we have soil respiration. Again, the colors are the same as before. And so you can see that particularly at the highest rate, at the 4.5 dry ton, we are seeing an increase in microbial activity, but also the amount of food available for the microbes. Again, I plotted these on the scoring curves for the active carbon. You can see that the non-biosolids treatments are in the medium low scoring range. The 2 middle biosolids treatments are in the medium, and at the 4.5 dry ton, we're getting into the high scoring range. So again, if you plot this on the scoring curve, you can see that we're having an effect on these biological soil health indicators. Here I'm also showing the soil protein nitrogen pool, and we see a similar trend with the highest in the 4.5 dry ton. And here we see that the 3 dry ton does actually have a significant increase. When I plot these on the coarse scoring curve, they're actually quite low. This may be due to the fact, as I mentioned, that biosolids had not been applied for 4 years. So, we were coming up for an application. This is also a fairly new indicator, so there's not quite the data that is available for some of the other indicators, particularly for the Western region. So I want to emphasize also that potentially mineralizable nitrogen showed the same trend as the protein. And also that there is really a need to improve these soil health scoring curves to be able to apply them to the soils and the systems that we have in the Western United States. As I mentioned, a lot of these are based on data in the Midwest and the Northeastern US, which really have different soils than we do. So this is some of the work my program is doing alongside this biosolids work. I also want to mention that the Soil Health Institute did also measure total microbial biomass, and we did not see any significant differences in total microbial biomass, nor did we see differences in the carbon cycling enzymes at this site. But I do want to emphasize that this was a one-time point snapshot, and these tend to be more temporally variable measurements. And so it would be interesting to look at other time points other than this spring time point to see whether we pick up differences. And a few chemical properties, soil pH on the top there, you can see that we're seeing quite a decline in soil pH in the highest biosolids treatment, below 5, around 4.7. And we also see a numerical decrease in soil pH with the 3 dry 10, though it's still around 5. So this could potentially be an issue for things like aluminum toxicity. We also saw an increase in soil EC, though these are all still within the normal range for electrical conductivity, so not a concern there. So the Soil Health Institute, for all of their sampling, measured the top 6 inches, which is typically what is used in a lot of soil health assessments. But in this no-till system where there's biosolids being applied to the surface, I was curious how these properties differ at different points, as I mentioned. So we measured 0 to 2, 2 to 6, and 6 to 12 inches, and measured some of the same properties that were measured in the Soil Health Institute project. So here I'm showing the active carbon pool. Again, that's the microbially available carbon in the soil. And you can see that the differences in the treatments are really just concentrated in the soil surface and not as much in the deeper depths. With soil respiration, we see similar trends, though there were some differentiations between the treatments in that 2 to 6 inch depth, but no differences at the 6 to 12 inch depth. For soil protein, again, we see the biggest differences just in that top 2 inches of soil. And soil pH is, a different one. We see much lower soil pH in the surface soil, so in the top 6 inches. And the soil pH for all of the treatments is quite a bit higher, close to 6.5 to 7 in that 6 to 12 inch range. So you can see that the fertilization over the last 24 years has had an effect on soil pH. And we do see differences in the different treatments. So the no fertilizer has the highest pH of all of the treatments, and then we see progressively lower pH as you add fertilizer, and then increasing rates of biosolids. And then I'm showing here Olsen phosphorus, which I didn't show in the 6 inch range, but I wanted to show it here because we really see that there is accumulation of phosphorus in the top 6 inches with all of the biosolids treatments. For Olsen phosphorus, we usually categorize 25 to 50 milligrams per kilogram as being high. So you can see both the 3 and the 4.5 dry ton biosolids are in that high range. And then the 2 dry ton is in kind of a medium range. So, nothing too concerning. We're not getting excessive here, but it is something to keep an eye on, which is also why biosolids are not applied to this site, every year or even every other year. So I haven't talked at all about yield yet, but here's some yield data from 2019. So this was the year right after these samples were taken. And you can see that there's quite a difference in yield between the biosolids and non-biosolids treatments. So the no fertilizer, not surprisingly, has quite low yield. These were oats planted this particular year. We see kind of medium yield with the synthetic fertilizer, and we really do see a benefit of having biosolids, but there isn't a significant difference between the biosolids treatments. So in terms of yield, you can do well adding 2 dry tons of biosolids per acre. You might not get the same benefits in terms of water holding capacity or active carbon as you would get at the higher rates, but in terms of yield, you still are going to get a benefit over the synthetic fertilizer. So just to summarize, again, we wanted to look at the impact on physical, biological, and chemical soil health properties. The higher rates of biosolids increase available water holding capacity and decrease bulk density, which in this system with low rainfall, coarse soils, that water holding capacity is going to make a big difference. We also saw in terms of biological indicators that active carbon and respiration were higher at the highest biosolids application rate, that 4.5 dry ton. Soil protein was higher with both the 3 and the 4.5 dry ton rate. but we didn't see any differences in microbial biomass itself at this sampling point. Soil pH decreased below 5 at the highest biosolids rate, and electrical conductivity, which is a measure of salinity, increased, but it wasn't agronomically significant. And then in terms of how these properties vary with depth in this no-till system, we saw the largest effect of the biosolids at the soil surface, with medium effects at that 2 to 6 inch range, and then very little effect of the treatment at the 6 to 12 inch depth. So with that, I would like to acknowledge particularly the Soil Health Institute for working with us and choosing this site as part of their project. And we're very happy to have all this data looking at how biosolids have affected these soil health properties, and particularly Shannon Capolazzi, who came out and sampled with us and sampled all over the Western US for this project. Thank you also to Boulder Park Incorporated, who helps us apply the biosolids, particularly Dave Rude. Douglas Poole, the grower, who's allowed us to continue this long-term site, which is so valuable over the last 24 years. And Jake Finlinson from King County, who works with us closely. And also thank you to our funding sources, Northwest Biosolids Research Foundation and the King County Department of Natural Resources and Parks.